Section 13.4 talks about the T cell response within the specific immune response. And we're going to look at the three major types of T cells and then their main functions. All right, so T cells are the cellular component of the third line of defense. B cells are going to be classified as the humoral immune response, the humoral component of the third line of defense. And I may not have mentioned that, but it's humoral and cellular. Humoral is going to be B cells. Cellular, even though B cells are cells, the cellular component of the third line is going to be T cells. So what you have in this table are a short description of the 5 major types of T cells, but really what it breaks down to is that you have T helper cells, the cytotoxic T cells, and then T regulatory cells. And all of these cells, based on the delineation here, are going to have a specific function. So here you have, in the first 3 lines, you have T helper cells 1, T helper cell 2, and the T helper cell 17. All of these are going to be classified as CD4 positive. All right, so those express the CD4 co-receptor for the T cell receptor. And as their name indicates, they're going to help the immune response. All right, they're going to help by activating different components of the immune response. And on the right side there, it shows you specifically what they're doing. All right, whether they are activating the cell-mediated pathway, whether they are activating or helping drive B cell proliferation, whether they are promoting inflammation. And to do that, they're going to secrete specific chemicals or interleukins as indicated by this IL, IL-4, IL-5, IL-13, IL-17. All of those different chemical mediators are going to result in their specific function. So they're going to help the immune response. And if you think about it, when we talk about HIV and AIDS, right, what happens there? Well, the HIV virus is actually going to specifically attach to the T helper cells. And over time, the T helper cells, that population is going to be driven down and killed off because of the virus. And so what happens when a person officially becomes, moves or crosses over from being HIV positive to having AIDS? Well, AIDS is a syndrome, so it's going to be a variety of different diseases, but they come down with kind of oddball infections or oddball cancers, those that aren't seen very often. And they're not seen very often because our immune system fights them off. However, with HIV, you're taking out all of these T helper cells. And so you're essentially wiping out a person's immune system. Because if you don't have the helper T cells, you really can't maximize your T cytotoxic response or your B cell response. And so then that's why a person with AIDS has different types of meningitis, has a major yeast infection that moves throughout their entire body, which generally you don't see that in immunocompetent individuals. But if they're immunocompromised severely with a lack of T helper cells, then they come down with those different diseases. All right, then T regulatory cells. These cells are also CD4 positive, and this group of T cells has only been officially around, because that's when they were identified, maybe 20 or 30 years. And 30 years is probably pushing it. But these are regulatory cells, all right? They're part of the T cell family. They express CD4, and what they're going to do is help regulate the T response, all right? So control specific immune response reactions, prevents autoimmunity. And that's very important. Then the last subgroup are the cytotoxic T cells. All right, and these cells are going to be CD8 positive. And that's an easy way to differentiate the T cells. Are they CD4 positive? Are they CD8? A functional T cell is never going to have, or I should say a mature T cell, is never going to have both a CD4 and a CD8 molecule on its surface. Some of the immature stages, as the T cells are developing and maturing, right, they will express both CD4 and CD8, and those are going to be called double positive T cells, but that's only like, adolescent age, adolescent years type expression. And not in just kids, but in the maturity process of the T cell. They've just been formed, and they leave the bone marrow to go to the thymus, and they start their maturation process there. All right, that's at some point when they're in the thymus and they're in that maturation process, they will be both CD4 and CD8 double positive. All right, so helper T cells, kind of already mentioned this, right? They're going to help regulate the immune response to antigens. All right, they're going to activate macrophages, and they do this by secreting different interleukins, all right, those IL molecules. And they actually make up a pretty large portion of the T cell population. And that's really all I want to say. We aren't going to differentiate between T helper 1 and T helper 2, even though if you have a particular disease, those are, that would be something that a clinician might do, is to actually determine what your subset of T helper cells is. All right, the cytotoxic T cells, those are cells that kill other cells, right? So their cyto is death, toxic, death, cyto is cell, toxic is the death part, so they kill cells. And the cells that they're going to target are going to be viral infected cells. So they're going to recognize that a cell, whether it's a throat cell or maybe a liver cell, whatever cell it might be, is infected with a virus, and part of that virus is going to be put on the cell surface. Actually, in some virus-infected cells, the cell itself can express a molecule on its surface, kind of like a flag warning the immune system, things are not right here, come take us out. Because then if the cell is taken out when it has a virus in it, the virus can't finish the process of maturation, and then the viral load will decrease. All right, these cells will also take out cancer cells. And so our cytotoxic T cell population is actually constantly moving through our body looking for abnormal cells. And if an abnormal cell is found and recognized as abnormal, the cytotoxic T cell will kill it. And that's important because if that cell isn't killed off, then it can replicate and become more and more, and then you end up having a tumor or you have a widespread cancer. And then also cells from other animals and humans, all right, the most important factor in graft rejection. All right, so if you have an organ transplant, and I mentioned this in maybe it was section 3 or section 2, right, if you have an organ transplant and your immune system can actually recognize that as foreign and will try to reject it, try to attack it, because it sees it as foreign. That's why, again, those organs have to be matched properly. All right, so here is table 13.5, looking at T cell activation. And you can simply look at this cell or this picture on the computer screen. If you can blow this up a little bit. All right, you have the dendritic cell there. It's called an antigen-presenting cell in this context, but you see all the little dendrites, all the finger-like projections from that cell, so you know it's a dendritic cell. And the dendritic cell presents the processed antigen to the T cell, and this is a CD4 T cell, all right, the MHC class II molecule, the antigen peptide right in there. And then the CD4 cell is going to be activated. And so it's at this point where it can start to, or it can differentiate into these different types of T cells, and then it can continue to do its job. So the T helper 1 cells activates macrophages. Not that you need to know that, but there are specific rules for each of them. All right, produce IL-4 and other B cell growth factors. All right, so that it can activate this B cell. The B cell will then go on to be either a regulatory B cell, a plasma cell that secretes antibodies, or a memory B cell. All right, and then down here, you have the cytotoxic T cell activity, where same scenario. And then this, however, is not a CD4 positive T cell, but a CD8. It sees the antigen, but it's also going to receive chemical signals in the form of IL-2 by the T helper 1 cell that helps to promote activation of the CD8 positive T cell, and then it can become either a memory T cell or activated and go on to kill cells. All right, so this is an infected host cell, and you can see these perforins. Those are secreted by the T cell, the cytotoxic T cell, goes directly to this infected host cell, and it's very close proximity here. You see that the two are going to be connected, and then this host cell that's infected with something is going to die. All right, so concept check. Cytotoxic T cells are programmed to kill all of the following except what? And your options are cancer cells, virus-infected cells, bacteria, foreign cells grafted into the body, or none of the choices is correct. All right, and the correct answer is bacteria. All right, cytotoxic T cells are not designed to kill bacteria. Those are what your phagocytes are for. All right, your macrophages, your dendritic cells, your B cells, neutrophils, all of those cells that are going to be designed to specifically attack bacteria and to phagocytize them, pull them in, and kill them. Cytotoxic T cells are not part of that group. And that is section 13.4.